Dear distinguished guests and media friends, good morning to the Americas, good afternoon to Europe, and good evening to Asia. On behalf of TAITRA, Taiwan External Trade Development Council, the organizer of this event, I would like to welcome you to the Taiwan Biotech Solution for COVID-19 testing kits live stream. The COVID-19 pandemic has had a massive impact on people's lives all around the world. With many tragic consequences, it has also had a profound impact on the global economy. Taiwan, learning from our experience in fighting SARS in the year 2003, has remained on constant alert and consequently took proactive measures to prevent a large-scale epidemic of COVID-19. As a result of COVID-19 crisis, the entire world is struggling, and Taiwan is eager to share our capabilities and experiences in order to help, cooperate, and partner with you in the search for more biotech solutions. Taiwan has extraordinary biotechnology capability in research and development. Academia Sinica, Taiwan's leading research institute, has discovered antibodies that could be used to develop a 20-minute rapid coronavirus detection kit. Taiwan's National Health Research Institute and National Defense Medical Center jointly unveiled a prototype of a rapid diagnostic test for COVID-19, through which test results take only 15 minutes. About 15 minutes, both institutes are also speeding up the process of developing vaccines and expect to have inspiring results soon. In addition to this, Taiwan has a number of outstanding high-tech bio companies, and it is my great pleasure to invite 7 of them to present here today. In addition to this, Taiwan has a number of outstanding high-tech bio companies, and it is my great pleasure to invite 7 of them to present here today. They are all dedicated to the development of high-quality test kits for COVID-19 and will receive FDA and CE certification very soon. I have no doubt that they will be able to offer reliable solutions for your countries. Before I go, I would like to thank you for your time and participation. We sincerely hope that TAITRA can help to connect you with leading Taiwanese companies for seamless cooperation on COVID-19 testing kits. And let me say finally, Taiwan can help and Taiwan is helping. Let's fight COVID-19 together. Thank you.
Hi, everyone. It's my pleasure to be here. Yes, I'm currently the president of National Taiwan University here in Taipei, Taiwan. And also I was the former coordinator of the Taiwan SARS task force back into 2003. Later on, I served more than 6 years as the Director General of Taiwan CDC. Through that experience, actually, I can contribute a little bit to the developments and overhaul our disease control systems right after the SARS. Now, this evening, I was asked to give you some short story about our fighting of COVID-19 during the last couple of months. Yeah. as you probably know, that Taiwan identified the first COVID-19 confirmed case on January 21st. One day after we activate our national command centers, and 5 days after we sent to a medical officer to Wuhan to do a site investigations. That show you how swiftly we respond to the informations that we get from the mainland China. Actually, that is the key elements we learned from the SARS outbreak in 2003. You need to move very, very fast, to fight any epidemic. Now, since the first case we identified in the late January, today we have only 429 cases as of I'm speaking today, and most of the cases are still imported cases. Only 55 cases are so-called the local or indigenous cases, which means that we don't have a widespread, sustained community spread, and with only 6 dead. If you're asking whether or not we have undetected cases or silent cases, because we know nowadays there are so many asymptomatic cases, I would say you bet. We do have a undetected symptomatic case, but I strongly believe the case number will be very low. Otherwise, there will be a sustained case, community spread. And so we will pick up the mortality or the hospitalization a lot. So, but we didn't see anything. So the idea is that because we move so swiftly, and so we are able to contain any small outbreak in these countries. And so we are sort of lucky to seize opportunity in the very beginnings to stem out any local transmissions. And so why Taiwan a success story? A lot of people ask me when I interviewed by PBS, CNN, and other news media around the world. And I would say it's probably is a combination of speed and solidarity, technology, trust by the public and the leadership, and probably some luck. Yeah, but I would say the speed is probably the most important things. You need to move very fast to make the decisions. And in Taiwan, we didn't undertake draconian control measures like city lockdowns or a blank orders for stay at home. Actually, so nowadays only shop close is the nightclub. Otherwise, people living is quite ordinary. No schools closing, no restaurant closed. We actually asking people, to practice a good personal hygiene, wash your hands, cover your mouth with mask and that sort of things. But the daily life is still going on pretty good, and so the economy is not that severely impact. And the reason we can do that is really we can do the testing and then identify cases very robustly. And then we do the contact tracing with the help of the modern technology and that sort of thing. Now, when we talk about the testing, a lot of people will say Taiwan probably not taking as much as what has been done in the South Korea or to Germany, that they do the mass screening, a drive-through testing, and so how can Taiwan do this? And why Taiwan doesn't conduct the mass testing like other places in South Korea or Japan, so not Japan, Germany, or like the Russians. And my answer is really kind of simple, because we have been doing this so-called precision public health. Actually, we don't need to do the mass testing. But saying so, that doesn't mean that Taiwan doesn't have a very distinguished and strong biotech company. As a matter of fact, the superb capacity of our bio company, testing included, is overshadowed by our outstanding achievement by our public health force and the cooperation from the public side. So today is a very kind of a unique opportunity to show you that the other side of Taiwan, our strong biotech companies. And I know they are strong precisely because, I'm the president of National Yangming University, which is the second best, if not the best, medical university in this country. And we have been cultivating thousands and thousands of high quality biotech human powers in the past 20 years. And also because over the last couple of years, we understand, if you want to have a strong public health, you need to have to work with the private sector. And that's why we have this very strong biotech company, including the testing capacities. Now, during this global health crisis, I think Taiwan means something else. TAIWAN. That means take actions instantly with accurate number. That's Taiwan. And how can you get the accurate numbers? Do the quality test. And so without further ado, I would like to bring our colleagues from our super biotech company. Thank you very much for your attention. Thank you.
Good morning, everyone. This is Dr. Yi. It is my pleasure to introduce you the 10 minutes Vstrip COVID-19 antigen rapid test. Next slide, please. After infection, viral RNA and the viral antigen start appear, followed with the immune response of the IgM and the IgG. The Vstrip COVID-19 antigen rapid test detect the presence of the SARS-CoV-2 antigen directly from the nasopharyngeal specimen. This Vstrip antigen rapid test will give you the most reliable result from the beginning to the end. Next slide, please. Current available tests for the detection of the COVID-19 disease are either viral RNA or DNA PCR test and the blood antibody immunoassay. The PCR-based test detects viral antigen with high sensitivity, but the tests are required in the equipment dependent and need a trained technician, high cost, and a longer turnaround time. On the other hand, the blood antibody test is easy to operate with quick turnaround time, but the test detects immune response, IgM and IgG, for the status of the patient after infection. Therefore, the Vstrip COVID-19 antigen rapid test really combine both advantage of these two tests into one and give you the best result in 10 minutes. Next slide, please. It is fast and simple to operate the Vstrip COVID-19 antigen rapid test. Only 3 steps involved. First, collect the nasopharyngeal specimen secretion. Second, extract the viral antigen in a pre-dispensed sample vial. The third, insert the test strip into the sample vial and read the result in 10 minutes. 2 lines on your right indicate, especially with the red line on the bottom, indicate that the viral antigen is presented in the specimen. If only one control line shows up, means negative. If control line does not show up, means the test is invalid. Next slide, please. This is the analytical performance of the Vstrip COVID-19 antigen rapid test. On top of the chart, you can see the analytical sensitivity of this test. The limit of the detection is TCID 50 is 79, which just means it's extremely sensitive of the test. From the 6 bacteria and the 13 viruses respiratory-related disease panel, no cross-reactivity was found from any of these microorganisms, which means the test is highly specific also. Additional table on the bottom also show that no interference from those commonly used drug and the whole blood also. Next slide, please. This product is a collaboration from the following 3 companies. And from the antibody design to the scale up and the purification of the antibody, eventually down to the optimization of the test. So in summary, we have successfully developed the sensitive and the specific 10 minutes COVID-19 antigen rapid test. Currently, we have submitted this test to the FDA for emergency use authorization. And hopefully, we all can use this test to manage it and map out the world COVID-19 pandemic disease in the very near future. Thank you very much.
Hello, all business partners from all over the world. I think we are now here together because we have the same purpose. We are looking for the best solution for detecting the COVID-19 virus and fight it. How to choose the correct detection kit in this moment is very important. We don't have any more chance again to do mistake again. Next slide, please. So there are 3 kinds of detection kit in the market. So let me introduce to you. The first one is the real-time PCR system. The other one is rapid test. Real-time PCR is the golden standard to detect the virus. No doubt, because this was authorized by the WHO and all the scientists, and the virus have the golden standard is a real-time PCR standard. And rapid test is used in different applications based on the different requests. Next slide, please. To compare within real-time PCR system and rapid test, and I think that is the necessary way to let you know our workflow. We had optimized the procedure of our real-time PCR from many steps to 1 step. Total times to run our real-time PCR only about 50 minutes. I think you will agree this is a little bit times than the rapid test. Okay, next slide, please. There are 3 important points how to choose a real-time PCR. Yeah, real-time PCR, so many products, real-time PCR for COVID-19 in the world, but how to choose it, this is very important. Okay, there are sensitivity. specificity and limit of detection is very important. And then how Kimforest developed this detection kit with a high sensitivity. Okay, let me tell you. Up to date, there are about 2,865 COVID-19 sequencing has been reported in GISID database. We do alignment and mapping our probes to this database by in silico computed technique. And we then develop a detection probe in our product can recognize all of the different types of the virus, such as Asia type, China type, Europe, and Asia, USA type. So that means we can detect all the different COVID-19 virus with this kit. So this is a high sensitivity. Next slide, please. The second point is about the specificity. In order to match to EUA FDA guidelines, we use the same in silico computed technique to verify our detection probes. I'm proud to tell you this result of this specific design is no cross reaction with other common respiratory flora or pathogens. That means only SARS-CoV-2 virus will be detected in our product. No one can doubt if I say this is a high specificity product. Next slide, please. Next slide, please. Okay, so let's talk about the last important for real-time PCR technical. Limit of detection is very important. The limit of detection of our product is less than 10 copies of virus cell. That means even less than 10 or 5 copies of the virus can be detected by our product. Okay, let me summarize the powerful of Kimforest SARS-CoV-2 detection kit is high sensitivity, high specificity, and less of 10 copies of virus can be detected. ROD is less than 10. Next slide, please. No, up. Too fast. Another slide. Yes. Most of the user have their own real-time PCR machine. If the user use the same real-time PCR machine, what we suggested on our protocol, step 1 plus, this machine is made by Thermo Fisher. We can offer them the automation report output software. If they didn't, okay, don't worry. We also can support them how to output their detection report. Okay, next slide, please. Our product, Kimforest for SARS-CoV-2 detection kit, is manufactured in GMP factory and quality control by ISO 13485. We already got CE conformity, and the US EUA will be soon registration finished. So this is a good choice for you. Next slide, please. Okay, from all of all, just to remember, Kimforest, you will get the best solution for detecting COVID-19. Together, we fight the virus. Thank you very much.
Hi, everyone. Thank you for joining this evening tonight. A quick background about our company. General Biologicals Corporation and we have been focusing on hepatitis, which is a virus and other tumor markers in immunoassays, molecular diagnostics, and a few rapid tests. Our main focus is about the COVID-19 PCR kits. So from the slide, you can probably read most of the information on there. So basically, we offer two kinds of tests or kits. One is to target genes, and one is just two based on the countries and the pricing, each customer's or partner's needs. We all offer both, and then each kit has 100 patient tests per kit. And we do work with several PCR kits. For example, the Antigen, ABI, and Biotech. So next, please. We have been manufacturing these kits, well, these two kits for a while now. So we just recently got the CE mark. And as I mentioned before, since we were founded in 1984, so we do have a capability to scale up the manufacturing capability. For example, for the test we are offering right now, we can potentially offer 800,000 kits per month and up to 1.6 million. But doesn't mean we have 800,000 kits tests available right now. It just depends on the orders. As you know, we've been selling overseas, not just domestically, but internationally over the world. So we do have the required ISO 13485, been audited by the European and as well as Korean for GMP certifications, as well as quality systems. On the right-hand side, there's the pictures of our kits. Next, please. So product features. Vice President Tsai actually mentioned the most important key things in the last talk. We do offer pretty good LAD limit detections, and we can offer 3 or 2 target genes, as well as working with simultaneously different brands of PCR machines. So it's pretty much open system. We test them out on the one I mentioned. We do also have the extraction kits that go with it. So it's just not the kits we're selling, it's actually the whole solution we are promoting, depending on the customer's needs. One key thing here, what we brought up is the CV, coefficient evaluation. From the manufacturing perspective, consistency is very important. So since we've been manufacturing class 3 hepatitis or virus, for example, we are confident to provide great consistency for the kits. Last but not least, we also include the IC internal control in our kits. Next, please. Oh, by the way, for all the performance data, details, if you need them, I have my contact info in last slides. I just thought I can give a quick summary on the products we offer right now. Also in the pipeline, we also working on the rapid test and ELISA kits. As I mentioned before, we've been manufacturing massively those 200 kits for the past years. I don't have the details right now from me right now, but if you do want the details, please do send me an email. And we are, just estimation, hopefully to have a pilot kits ready by early May, and hopefully goes to production toward the end of May. With ELISA kits, we actually are moving more or faster, so we're launching that toward the end of May. Next, please. As a summary, we've been manufacturing, as I mentioned, actually a few times before, since 1984. All the way from the platform IA, the Asian platform, ELISA, RT-PCR, rapid test, and just like COVID-19 is actually class 3, high risk. We manufacture, specialize in hepatitis, which are virus tumor markers, and we have been also partnered with the pharma, developing companion diagnostic products. So I'll be happy to send more information about that. And one key thing is about our company is we are not just a manufacturer. We also have 2 clinical labs in Taiwan and the US. that actually run the test. So we have technicians that actually understand on the running perspectives, not just the product features. We actually work with them on systems, so they know what's the protocol setup, and what's the best way for our kids to work with the systems. And as far as compliances, we pretty much certify with the ISOs and required to sell domestic and overseas. So next slide. If you have any other questions, next slide, please. That'll be my last slides. Please do email me down there, or just check out Facebook. I'll be happy to send you more info. And I was reminded to mention this again. Our product has been CE marked, not certified, cannot say certified, so CE marked. And we are working on the FDA, UA. And what else? Oh yeah, in Taiwan, yes. So that's pretty much my presentations. Thank you. Any questions, please email me. Thanks.
I'm Chen from Leadgene Biomedical. Thank you for your join, and also thank Tetra for holding this meeting online. And it's my pleasure to have this opportunity to introduce our products here for you. Next. First of all, I would like to make a brief introduction of our company. We focus on the development and application of antibodies, proteins, and immunoplatforms. We are ISO 13485 and GMP certificated. And based on our technology and manufacturing plant, we offer not only high quality raw materials, but also final IVD products like rapid tests and ELISA kits. Next, thank you. For COVID-19, we dedicated ourselves to develop and generate materials for IVD and research use since the beginning of the outbreak. And now we have comprehensive product lines for different antigen and antibodies. Continuously, we finished the development of antigen rapid test, and it is undergoing the clinical validation. And following, we will have antibody rapid test and antibody ELISA kits in next month. Next. For our antigen rapid test, it contains 20 tests in a box, and each test has a sampling bottle and a testing cassette. And next. It is quite easy to perform the test. You can just put the swab into the sampling bottle and mix with the buffer, and just add 3 to 4 drops into the testing cassette. And the result can simply be read in 10 to 15 minutes. Next. Although our products are undergoing the clinical validation, but it showed great performance in preclinical data. Our test can recognize SARS and the SARS-CoV-2 and without cross-reaction with MERS and other coronaviruses. And it shows great sensitivity. It can reach nanogram per meter level. And it tested by recombinant proteins. And we also confirm our test by virus-like particle. Next. And finally, I would also like to introduce our high-quality antibodies and antigens for IVD manufacturing and academic research use. We have various materials, such as human anti-SARS and SARS-CoV-2 protein antibody, and different recombinant proteins of different coronaviruses and different viral proteases, which can accelerate the development for the knowledge and the application in diagnosis, prevention, and treatment. Next. And that's all my presentation. And let's work together to fight for this pandemic. If you have any questions or inquiries, welcome to contact us. Thank you very much.
Thanks to Tetra for organizing the meeting. Next slide, please. I want to start with this slide, which gives a picture of the onset of the virus infection that results in antibody production in humans. Antibodies are only produced many days after the infection. Therefore, an antibody test only can confirm a patient probably around 7 to 9 days after the virus has entered the body. What we really need is a more sensitive method to detect the virus at an early stage. That's why RT-qPCR is the preferred method. However, if you look at the picture carefully, you probably can only detect the presence of a virus at day 4 using RT-qPCR. What we need is really a more sensitive way of detecting virus much, much earlier. Most importantly, what Quark Bio wants to do is to make sure that recovering patients doesn't have any virus when they leave the hospital. We want to monitor the viral load carefully. Next slide, please. Our solution is a longitudinal viral load monitoring assay based on a proprietary dual-function digital quantitative PCR system, which can run both digital and conventional qPCR. On the left-hand side is our chip, which is the Q chip. This is a chip-based qPCR solution. And on the right is our thermocycler, which runs the Q chips. In the next few slides, I'll give a quick introduction of the company and the system. Next slide, please. Quark Bio was founded on the vision of using the most innovative technology to provide the highest quality precision healthcare solution to patients around the globe. This is what gets us up in the morning and pushes us every day to find better, faster, and more accurate solutions. Our team of 80 experts have committed hundreds of thousands of hours over the past decade into research, testing, and development to redefine what highest quality precision healthcare means to everybody and how it can make accessible to people around the globe. We're currently private and with about 50 plus patents. Next slide, please. Our mission is quite simple. We want to enable precision medicine through novel diagnostic technology and assay. Next slide, please. Therefore, we built the enabling technology, which is a chip-based qPCR digital PCR system I mentioned earlier. Because it's a chip-based qPCR system, we're able to accommodate hundreds of biomarkers, which means we can detect multiple genes at the same time. In addition, we also have the digital PCR functionality, which allows us to develop many different sensitive assays. And a very major advantage that we have is that this is a chip that's delivered ready to use. And when it gets to the user's hand, all you have to do is a few simple steps and you can run the reaction. In the next slide, there will be a video showing you how this works. Next slide, please. Can you play that? Thank you. Okay, so basically, we're preparing the sample right now. The pipette is automatic. We're sucking up the samples and loading it onto the chip. About 10 seconds, everything gets all the samples gets into the chip, placed into our PCR machine, and in less than 2 hours, you receive your results. Very simple operation and affordable. Next slide, please. So with this technology, we have utilized our digital PCR functionality to develop our SARS-CoV-2 assay. Because it is based on digital PCR, providing absolute quantification, the assay is ultra-sensitive, which, as I mentioned earlier, it's perfect for longitudinal viral load monitoring because of its precise quantification and low copy detection. Next slide, please. So this slide basically illustrates that this assay has great dynamic range. We can detect very low copies or very high copies of that virus itself by switching between digital and qPCR mode. Next slide, please. This slide demonstrates that the probe we designed has good specificity, meaning that we won't accidentally detect something that's non-coronavirus related, reducing false positive rate. Next slide, please. And this is our instrument sensitivity. Comparing to standard or conventional qPCR, we are 3 to 10 times higher in sensitivity. We can detect as low as 3 copies of virus presence. Next slide, please. Basically, we are in the process of applying for TFDA and FDA UA, and hopefully we'll receive the certification within a month or two. At this time, I just want to end this presentation by thanking you for your time. Hope we get through this pandemic together, and hug your loved ones, and stay safe and stay well. Thank you.
Good morning, good evening, and good noon for everyone participant. Thank you for this opportunity to have me here to present for what we have done in the past years. Actually, says it's the last 3 months. Yeah, this is based on what we have done before and to modify it into to fit the SARS-CoV-2 detection. Next slide, please. And this is a short introduction, device summary of ours. Not really the prototype because we are now we are going into the manufacture phase and the clinical validation phase now, and seeking for the collaboration with everyone for the interesting partners. And this is our system specification. The maximum test sample is around is 6, and the sample volume is 50 microliters, and this is the capillary tube here, and we use the triple blue LED array and the photodiode array to make this simple and very light. You can see that the weight is less than 600 grams here, not including the battery. And if you add on the adapter, that's only about 680 grams only. Okay, next slide, please. And this is our device summary. You can see that's very simple. That you put things here into the machine, and after 20 to 30 minutes, then you will see that the positive side and the negative side here. So that's very intuitive here. And let's design for the molecular diagnostic for the qualitative usage. In the old days, we use this to develop for the influenza, so we modify this into for the SARS-CoV-2. Next slide, please. And this is our technology summary. Because the enzyme we designed by ourselves to fit for the faster convection speed here. So let's say faster tag is especially fast, and it is highly tolerant for the not contamination for the sample. So you can input more samples here to increase its sensitivity. And the other modification for this is what we done is to modify the tube shape and for the tube height and for the viscosity of the formulation of the reagent kit here. And next slide, please. And I think this is the last slide here. This is our patent status, including 3 sets of the patent, except for the last one, only issued. Yeah, thank you very much.
Hello everyone, I'm Aaron from TCI. Welcome to TCI. You can see here is our laboratory. You can see the encounter with the nanostream and a lot of device, and you can see the automatically device in our laboratory. The gene laboratory is already passed the good laboratory practice in the world. Next please. You can see we already certificate by ISOs and the GLP. Next, please. Okay, you can see the QVS 96S. Next page, please. The total examination process, you can see is normal qPCR procedure, but we use the robot to make it faster. when we fully automatic for 2016 per day in 24 hours. And if we want to test 96 samples, it only spend 3 hours. Next, please. You can see the spec data in this slide, including the dimension and the weights and the power and the air pressure. There's 3 stations. Next, please. You can see the 3 stations, including the RNA extraction station, dispensation station, and the fluorescence signal detection stations. Next, please. The first station is an automatically nucleic acid extraction device to extract nucleic acid from upper and the lower respiratory specimen, in which sample identities and recognized via barcode and the specimen tube. You can see the robot is doing the job. And then next, please. The next station is to extract the nucleic acid sample, automatically transfer aliquot and mix with the qPCR region. It actually is normal testing procedure, but we make it faster by robot, and it already certificated by Taiwan CDC. Why we say our QBS96 accuracy of the 99.99%, because it used by the qPCR machine. All our government and the whole while test laboratory, we are all use the same machine to test COVID-19, and it automatically transfer the mixture for the qPCR reaction. These 3 parts are fully automatically operation without operator actions. The testing results are exported via self-generation algorithm to combine the sample and the identity and the result. Next, please. And the next, please. You can see this is the result. We already passed the Taiwan CDC 3,000 sample pressure test, and it's 100% correct. Next, please. Please play the video.
World's most accurate coronavirus testing machine. Discover the secret of TCI. QVS96, the world's first fully automated virus scanner. Based on the qPCR detection technology, uses robotic arms and applies a unique specimen number to avoid false test results caused by human error. It takes only 50 minutes to obtain up to 96 test results, 24/7 comprehensive monitor, and able to detect viruses of various other strains through the conversion of the module, which greatly increases the detection flow rate. With high output, full automation, and zero contamination, QVS96 virus scanners can facilitate the works of disease control and quarantine departments in multiple countries to help curb the spread of the virus. TCI is here to help protect the safety of you and the people around you.
Hello everyone, this is Fong from United Biopharma, short as UBP. It is my honor to be here to introduce you the series product developed by UBI Group. So we UBP belong to UBI Group. So in this group, the different company has different expertise and focus, including diagnostic kits, vaccine for animal or human, and therapeutic protein or chemical drug. So from the beginning of this year, we have initiated a big project to work on the serious product against the COVID-19, which including the diagnostic kit, peptide vaccine, subunit vaccine, and protein drugs. So this is our first product as an ELISA kit, which was completed in March. And this ELISA kit is now listed on FDA website as being in compliance with their March 16, 2020 policy on COVID-19 serology test. So this product is already being used in US now. So the reason why we can quickly develop this COVID-19 ELISA kit was because that we had the experience of developing a SARS diagnostic in 2003. So from the data here, you can see a clear cutoff line can be shown here to differentiate the positive sample and negative sample. So by leveraging the experience of SARS ELISA, we developed this high sensitivity and specificity SARS-CoV-2 ELISA kit. We had tested this sample by specimens from Beijing, California, New York, and Taiwan. The positive sample, which are from PCR confirmed COVID-19 patients. Those samples are divided into 4 different groups. The first group is lower than 10 days infection. The second will be 10 days after the infection. And the third group will be discharged from the hospital and 14 days after the discharge. So from the first stage, as 10 days upon infection, we have very low detection rate from the patient. However, after 10 days of infection, we can all get the positive results. And we also test negative sample for the specificity test. So the samples from other strains of COVID-19 and other different virus infection, including HIV, HCV, and HBV, they all give us negative results. So that's why we think our system has very high sensitivity and specificity. So this is just to show you our ELISA system, which is suitable for large-scale application, either manually or by automated ELISA machine. Most labs have this capability to do so. So this can be done either internally or a large number of accredited labs operating in the area. So the format is 96 wells per plate and approximately 2 hours of incubation time. Manually, this translated to approximately 200 samples per shift. On an automated platform, one could proceed over 750 samples in 8 hours. So this is a table just to show you the different testing kits can be used in different stages. So at the early stage of infection, the viral load is relatively high, but the antibody is relatively low. So by using qPCR can give better chance to detect the infected patient. However, when human immune system worked, the viral load can be suppressed, and however, the antibody increased. So in this stage, the antibody ELISA kit may give better chance to detect the infected population. So that's why we considering our ELISA test it would be a good tool to detect community transmission in a large scale. So this is some highlight of our marketing and sales. Just like I mentioned, we are now listed on the FDA website, and CE mark is to be obtained in June. This diagnostic kit is manufactured and supplied from US and also being used in different states or cities in the US now. And this test should be performed in a well-equipped serology laboratory. The minimum order would be 9,600 tests, and the current manufacturing capacity would be 500,000 tests per 2 weeks. So other than the ELISA kit, just like I mentioned, we also developing the different products against COVID-19. If any of you are interested, we are open for discussion for the co-development. So thank you very much for your attention.
Hi everyone, I'm happy to have this chance to introduce a fully automated molecular assay GeneReach has developed for COVID-19. The demands for PCR detection of SARS-CoV-2 are dramatically increasing due to the fast spread of COVID-19. Currently, PCR testing is done mainly in central labs. The capacity of PCR detection has become the bottleneck in the medical systems around the world. In addition, for places with no direct access to lab testing, it can take days to obtain the test results. GeneReach's sample-to-answer packet-central PCR system can help decentralize testing and allow PCR done right at COVID-19's hotspots. This is a fast and deployable benchtop system. Results are automated interpreted to provide qualitative answers in 85 minutes. The Pocket Central COVID-19 assay has obtained the EUA from Taiwan FDA and has been CE marked. This fully automated molecular assay can run 1 to 8 tests per run. It involves minimal hands-on steps and offers direct result display. All solutions and consumables required for the assay are provided in 2 preloaded ready-to-use cartridges for each test. The PCR premix provides accurate results with internal control. Its lyophilized unidose format allows easy shipping at ambient temperature. The Pocket Central assay targets well-conserved sequences in the ORF1AB region and can be used to test oropharyngeal swab. It has been verified by both wet and dry tests to have great analytical specificity, not cross-reacting with close relatives and common respiratory pathogens. Testing of 70 contrived oropharyngeal swab samples showed that the Pocket Central assay and 1 real-time RT-PCR assay listed by WHO have equivalent clinical performance. Total agreement between the 2 assays was 98.6% with kappa value of 0.97. Further validation with 30 clinical oropharyngeal swab samples at 1 hospital in Taiwan also showed that the Pocket Central assay had equivalent clinical performance with the hospital's method. The total agreement was 96.67%. The kappa value was 0.93. This fully automated PCR assay has been used in various countries, including Indonesia, Cambodia, UAE, and Pakistan. This is a news released by WHO in April. WHO donated Pocket Central machines and PCR tests to the Pakistan government to help their fight against COVID-19. The next slide shows a short video clip to demonstrate to you the simple workflow of the Pocket Central system. That's all. Thanks for your attention.
Hi, as my Twitter reaches 1 million followers, this milestone comes at a unique and unprecedented time in history. Social media helps me communicate with the people of Taiwan, but it also helps Taiwan connect with the world when connectivity has a greater meaning. The global battle against COVID-19 has changed life as we know it. The world is full of uncertainty about what the future holds. But if this crisis has taught us anything, it is this. Together, we can be more than the sum of our parts. The people of Taiwan have shown me this since day one. Our society has united and risen to meet this challenge in so many ways. Taiwanese medical teams are working day and night to ensure our citizens and friends abroad are better prepared to fight the coronavirus. Private sector manufacturers have dedicated their resources and capabilities to form Team Taiwan, allowing us to dramatically increase production of critical supplies such as face masks and protective equipment. At the same time, our government has received countless messages from citizens across Taiwan, asking how they can personally donate supplies and funds to those most in need in the international community. Taiwan is determined to do our part to help end this pandemic by donating supplies, cooperating on research, and sharing our experiences. We know that we cannot afford to stand idle when the world is in need. It has been my honor as president to witness Taiwanese people's selfless desire to give back to the international community time and again, despite political pressure. Taiwan wants to engage with the world, and social media gives us a free, open platform to enter the global conversation. Pandemics have no borders, but fortunately for us, social media transcends distance as well. Coming together and sharing our stories remind us that we are not alone. We will get through this and we will do it together. I want to thank you all for supporting Taiwan, for sending us strength and encouragement from all corners of the world. And I hope that by sharing Taiwan's experiences and successes through Twitter, I can also share with you hope for a brighter tomorrow.